I'm going to focus in on one particular autoimmune disease today, lupus, but some of what I'm talking about, the sort of the broad strokes, are applicable to other autoimmune diseases. So I'm going to be bringing you up to date on new findings in lupus and new targets. All right, so as you may know, we have big interest at BRI in rheumatic diseases, and I want to start by telling you a little bit about our rheumatic disease registry and repository. Maybe some of you actually participate in that. So we have 4 different registries for the rheumatic disease registry and repository. We have rheumatoid arthritis, scleroderma, relapsing polychondritis, and lupus, and they're headed up by Dr. Jeff Carlin and Dr. Jane Buckner, and Sylvia Passo, who, if you are a donor, you would interact with her. And these repositories began in the 1990s and were expanded in the early 2000s. The last one that we added was the lupus registry, and to date, we've shared over 300 samples with investigators both within BRI and our collaborators outside. So I'm going to be focusing on lupus today. And as you may know, lupus is a systemic autoimmune disease that is chronic and leads to long-term inflammation. Its prevalence is about 20 to 150 cases per 100,000 patients worldwide. And it develops usually between 15 and 45 years of age, and it's primarily in women. The gender distribution is 9 women to 1 man. So really a disease of women in their early adult to mid-adult age. And it's also more frequent in some racial groups, so African American, Asian, Latino, and Native American people are much more likely to develop lupus. And what we've learned in the last 10 years is that lupus is caused by alterations or changes in the immune system and in 2 different pieces of the immune system. And I think Dr. Campbell introduced this to you in his talk, which are the innate and the adaptive immune system. And I'm going to talk a little bit more about that. So this is a slide kind of showing you the 2 arms of the immune system. So up here in blue is the innate immune system. These are the first responders. When you're exposed to a bacteria or a virus, these are the cells that are going to come and say, wow, we've got something bad going on here, and they're quickly going to alert the adaptive immune system down here with this green arrow. And how they alert the adaptive immune system is by secreting these hormones that bring the adaptive immune cells to the site of the infection. And I want to, these hormones are also called cytokines, and you'll hear me refer to them as cytokines in my talk. I want to draw your attention to one of these, which is interferon alpha. This is an important cytokine in lupus. You may have heard of the interferon signature that many lupus patients develop, so that's where that comes into play. So then in the adaptive immune system, you have 2 different types of cells. You have the T cells that Dr. Campbell talked about, and you have B cells. And the T cells generally help the B cells, and they do that by actually contacting the B cells, like coming into contact with them, and they also secrete hormones which impact the B cell. And then the B cells are sort of one of the bad guys in lupus because we get activation. Whoa.
I told you.
We get activation of the wrong kind of B cells. These are B cells that start interacting with your own body, with cellular contents in your body, and you develop what are, we call autoantibodies that react to these cellular contents. and it's these autoantibodies that drive the disease, so in combination with the cytokines. So what causes lupus? The innate and the adaptive immune system, but specifically the cytokines, the hormones, and we call them pro-inflammatory cytokines because they inflame. They inflame cells of the immune system, they inflame the tissues where they're locally secreted, and that's a good thing if you have an infection. It's not a good thing if you have autoimmunity. And the other players are the autoantibodies, and as I mentioned, they interact with pieces of dying cells. So the contents of the cells, that's what these autoantibodies interact with. And so how they cause a problem in lupus is, so for example, in the kidneys, these autoantibodies end up clogging up the kidney glomeruli, and you get lupus nephritis. In arteries, the autoantibodies plus the inflammatory cytokines promote atherosclerosis and heart disease, which is one of the side effects of lupus. And then you can get neuronal problems due to the autoantibodies binding to neurons. So that's how the immune components are causing some of the symptoms of lupus. And so who gets lupus? We know that there are many components to susceptibility to lupus, and some of those are shown on this slide. So there are genetic components, environmental, there's immunoregulatory, hormonal, and epigenetic. So the genetic susceptibility, we now know, and this is one of the things we've learned in the last 10 years, is what the gene variation is that puts someone at greater risk of developing lupus. And I'm going to talk about this in more detail. We also know that there are environmental triggers that can exacerbate your likelihood of getting lupus if you happen to have, the correct gene signature. Those things are like UV light, viruses, smoking, and microbiome. This is another thing we've learned through the omics, is that the type of bacteria that you have in your intestinal tract can influence your immune cells. Believe it or not, you have immune cells that live in and near your intestinal tract, and now we're starting to understand how the type of bacteria that you have in your intestinal tract actually trains those immune cells. So that's something that we know can influence as an environmental factor. Then, of course, as I alluded to earlier, hormones play a big role in the fact that women are much more likely to develop lupus than men. There's also pregnancy flares, and we know that there are genes on the X chromosome that are involved. And lastly, we're starting to understand in the last 10 years that there are epigenetic changes. So epigenetic changes are where you actually get modification of different genes, and it affects how well they're expressed. And I just want you to remember one thing from this is IL-2. We're going to come back to that at the end as a new treatment for lupus. Okay, so I mentioned the genetic susceptibility. So this is a graph from a paper, and what it's showing is for these different autoimmune diseases, and we'll focus on lupus, what is the relative risk of someone who has a sibling with lupus compared to the general population? So what you can see here for lupus is that if you have a sibling with lupus, you're 20 times more likely to develop lupus than someone in the general population. So there's a big piece of lupus susceptibility that is due to the genes that you inherit. And we now know that lupus, for most people, is a complex genetic disorder. There are a lot of genes that contribute to it. And so let's come back to who gets lupus. And I'm going to overlay on this. So remember, here's your innate and your adaptive immune system that are playing a role in developing lupus. So now let's add where the genes are that cause susceptibility for lupus. Okay, so there are a bunch of genes up here that impact the innate immune cells. We've also determined that there are genes that are involved in some of the cytokines or the hormones that the innate system uses to talk to the adaptive system, and especially several of these involve the interferon pathway, which is really important in lupus. We know there are genes that impact how well the T cells work. And then, as you can see, this is a long list for the bad guys, the B cells. There are a lot of genes that influence how the B cells work and how it causes them to develop secretion of autoantibodies. And then another big piece is how the body clears these autoantibodies, because that is also contributing to what's happening in the tissues, the tissue damage, because there's poor clearance of these autoantibodies. So, what did the genetics tell us? You saw there was a huge long list. Well, they tell us that, first of all, almost all of these genes are involved in the immune system. So, it's just reiterating to us that the immune system is very important in lupus. It's really the foundation of this disease. And it also shows, you saw how I had the genes listed in sort of long lists, that they fall into pathways, and the genes have been very helpful to understand what are the pathways that are driving this disease. Where do we need to target the therapeutics that are being developed for lupus? And then another piece of information is that some of these genes are also cause susceptibility for other autoimmune diseases like MS and rheumatoid arthritis and type 1 diabetes. And that tells us that there are mechanisms that are in common between these apparently diverse autoimmune diseases, that there's some fundamental issues that are shared by them, and maybe those are also good therapeutic targets, because once you have a therapy for one that targets one of those, it might be something that could be applicable to other autoimmune diseases. Okay, so I'm going to talk about the B cells a little bit, because that's one thing that I've been working on. So here we have the B cell, and it's interacting with a T cell. Remember I said that they could bump up against each other and interact. And this interaction gets both the T cell and the B cell excited, and the T cell especially makes cytokines that influence the B cell. The B cell here you can see is binding the pieces of the dying cell, and then it's making the autoantibodies that are binding the pieces of the dying cell, and those make immune complexes which damage the kidney and the arteries, etc. This is the list of genes that we know cause susceptibility to lupus, that are found, that are expressed in B cells and function in B cells. And I want to draw your attention to one of these, BANK1. That's a gene that we've been interested in. And so this is kind of a circle. You have the dying cell, it binds to the autoreactive B cell, the B cell interacts with the T cell, and you get cytokine production and production of autoantibodies. So BANK1 is a susceptibility gene for lupus, and one of the things that we found is if we look at people, and this is a graph, each dot is a person. If we look at people who don't carry the risk version of the BANK1 gene, so those are the ones in black, and we count their B cells in their peripheral blood. So we take a sample of blood and we count the B cells. What we find is that between people who do not carry the BANK1 risk, the gene that causes risk for lupus, versus those that do have the BANK1 risk, which are shown in red, we see an increase in the number of B cells in the people who have the BANK1 risk gene. So this is going from low to high. So you can see the red individuals have more B cells than the black ones. This is looking in healthy controls. These are people who don't have lupus. So this is something that may be predisposing them to develop lupus if they have an environmental trigger, a virus or whatever. Now, if we go and look in lupus patients, we see the same thing. So the same kind of graph. Here are the control subjects in black. You can see their number of B cells in their peripheral blood. Now, if you look at the lupus patients in comparison to these controls, what you see is they also have a lot of B cells. So what we're learning is that the BANK1 gene may cause susceptibility to lupus because it increases the number of B cells that are developing in your peripheral blood, and that is modeling, predicting what we see in lupus. So this is one piece of what might be going on in lupus. Okay, so going back to the innate and the adaptive immune system, we're going to talk a little bit about what the therapeutic targets are. Okay, so one therapeutic target that's been developed fairly recently is a drug that is going to neutralize or inactivate one of the cytokines that the innate system uses to talk to the adaptive system. This is a cytokine that is a growth factor for the B cells. It's called BAFF, and a new therapy called belimumab or Benlysta has been developed to inactivate that. So that's a new therapy that is showing promise in some lupus patients. We also have a therapy that removes the B cells. As I said, we saw that with the BANK1 risk gene, there was a lot more B cells. In lupus patients, we see there's a lot more B cells. So there's a therapy called rituxan that removes the B cells. Then we have drugs that interact with one of the cytokines that's both produced by B cells, IL-6, but also the innate cells. IL-6 is one of those inflammatory cytokines that's going to get the immune system overactive and the tissues overactive. And then we also have a drug which you may have heard of, Remicade or Enbrel, that binds to another inflammatory cytokine called TNF. That's another cytokine that plays a role in other autoimmune diseases like inflammatory bowel disease. Then we have drugs that target the T cells and their activation. We have Orencia, which is blocking one of the molecules that allows the T cells to interact. And can't see this. Zolaznes, which is blocking the molecules that the T cells use to signal. And then I want to talk to you about a new and exciting therapy. It's really just at the ground level, but I saw this presented last year at a scientific meeting, and it was just published this spring, and I'm actually going to show you a little bit of the data. So there's a cytokine that T cells need to grow and to talk to each other. It's called IL-2. You remember earlier I showed you a slide about IL-2 saying that it gets modified in lupus and its expression goes down. So what's been shown is that if you give patients a very low dose of IL-2, it's very effective at treating lupus. So this is data from the publication. It just came out in April. Here you see a lupus patient who was administered low levels of IL-2. 15 days later, you can see this cutaneous rash is completely cleared up. So this is a graph, and I'll walk you through this. So this is looking at lupus severity. So the higher the number, the more severe the lupus is. And every time that the patient received IL-2, it's one of these gray bars. And what you can see is this line saying how severe the lupus was. So it started out being quite severe, and as soon as they got the IL-2, it went way down. You can see it stays down while they're getting the IL-2, and then it slowly climbs back up once the IL-2 is discontinued. So how does this work? Well, IL-2 is really important for one of the immune cells, T cells, that put the brakes on the immune system. And these are kind of T cells that people have put a lot of effort into understanding in autoimmunity because of their role as putting the brakes on. They're called regulatory T cells. And what you can see on this lower graph is every time the person got IL-2 in gray, this is counting the number of regulatory T cells in their blood. You see that it spikes up. So every time they get IL-2, their regulatory T cells go up. So the theory is that giving people low doses, lupus patients low doses of IL-2 is increasing their regulatory T cells and dampening down the immune response. So this is very early days. It's only been given to a few patients, but it looks very promising. And I think in the next 10 years, you're going to see this as a very promising therapeutic in lupus and other autoimmune diseases. It's also been used in alopecia areata and had very good effects there. And so I think this is really the beginning of looking at this for autoimmunity. So I'll just end by talking about what I presented today. So we know that lupus is a systemic autoimmune disease, and it has an immune basis that is comprised of both the adaptive, the early responder, the innate, the early responders, as well as the more specific adaptive immune system. And two of the big drivers are autoantibodies and cytokines, the hormones that the immune system uses to talk to each other. We know that the disease is impacted by various susceptibilities, so genetic susceptibility, environmental triggers, and hormones. We know that new studies have defined the SLE susceptibility genes, and these have told us some important things about the underlying immune system and underlying immune pathogenesis in lupus, that B cells especially and T cells are important, cytokines, I get a little carried away. There. Cytokines are important, and clearance pathways for the T cells. And at BRI here, we're interested in understanding how alterations in B cells are encoded by the genetics that cause susceptibility for lupus. And lastly, that new therapeutics have been developed for treatment of lupus, and I think are going to be applicable to other autoimmune diseases. So lastly, I'll just say thanks to those of you who may participate in our registries. It's huge. It helps us to do what we do here, which is to bench to bedside to bedside to bench to bedside. So I'll end there and take any questions that you have.
Does lupus develop, or does someone have it? How does lupus, how does someone have lupus? Is it something that develops like middle age, or?
So what we think is that there is a genetic susceptibility, so you're sort of at risk, and then when the appropriate environmental influences happen, that underlying risk then sort of moves forward and blossoms into lupus. That's probably why it happens a little bit later, especially with the effect of hormones being a key player.
Can that be said of many other autoimmune diseases, that there's a latent predisposition with environmental triggers or something?
Yes, that's true. Now, there are some autoimmune diseases like type 1 diabetes that can develop very early, and as Dan was referring to, that we're just starting to get a handle on the heterogeneity within a disease, and that's where these omics have come in to be so important. It's trying to understand the subtypes within a particular autoimmune disease and which drug is going to help these subtypes rather than just looking at it as a whole. So type 1 diabetes that affects children at a very early age may be a slightly different disease than type 1 diabetes that occurs, in an adult. And so I think we're going to start to get at that. But in general, there is a predisposition and then an environmental trigger kind of puts you over the edge, and you start developing, you may not even know it, but you start to develop some of the early players in the pathogenesis. And that's the other thing we're trying to understand is, through these omics techniques, can we develop biomarkers that can help us see when, you may not be aware that you're having symptoms, but, maybe you have a sibling that has lupus, and trying to understand, like, when you might be starting to exhibit features that may eventually lead to lupus and, at some point start to give people preventative therapies.
Has there been any research into why autoimmune disorders tend to affect women more than men?
There's a lot of interest in that, and I don't know, Dr. Buckner, do you have any?
Yeah, and I'll speak to it a little bit. It's a fascinating aspect of autoimmunity. Some of that has to do with hormones. Some of that may well have to do with the fact women have to deal with a foreign tissue in their body during pregnancy, so we're uniquely designed to tolerate that, but that may be part of the problem with autoimmunity. There are some diseases that predominantly affect men as well, and I think those are also help us get insight into these different mechanisms. Certainly an area of study in many cases. So, and I'll touch on that a little bit when I talk in the next few minutes.
Are there side effects, do you know, associated with taking IL-2 in most patients?
I think that there are. I mean, there's definitely side effects when you're receiving high-dose IL-2. I think you feel pretty bad, and Jane may be able, Dr. Buckner may be able to speak more specifically to what some of those side effects are. But the advantage of the low-dose IL-2, because it is a low dose, that a lot of the symptoms and side effects of high-dose IL-2 are ameliorated, so it's definitely much better.
When you have so many drugs that can impact different pathways, how do you decide which ones are going to help somebody? I mean, can you apply them all and hope for the best?
Well, I think that's where we're trying to go, is understanding, stratifying patients for therapy. Who are the patients that have expanded Bcells that may benefit from rituximab or the anti-BAFF theory, therapy, the belimumab, Benlysta. I know things by their lab name, not by the name of the drug that you actually take, as compared to someone who may have high levels of inflammatory cytokines. So I think starting, that this real focus on starting to stratify patients to understand who is likely to benefit most from individual therapies is really going to turn out to be beneficial.
What about patients who start on a particular drug and fail on that one and then have to start another drug? Has that been studied, how the immune system changes with those?
Yeah, one sort of unfortunate side effect of a lot of the therapies that have been developed for autoimmunity is that they actually use antibodies. So you remember we had the Bcells in lupus, they make autoantibodies. So how we neutralize cytokines, how we inactivate or remove Bcells is we put in an autoantibody that binds to those things and then it goes away. Well, the problem is that your immune system, in some people, your immune system starts to see those antibodies, the drug you're being treated with is foreign. And so you start to get an immune response to the drug. And so then what happens is the drug gets neutralized when it's being administered. And a lot of times that's why people will fail one of the therapies. And sometimes there's, like for TNF, there's multiple TNF drugs, and maybe you can try one of the other ones. But sometimes you just have to move on to, unfortunately, it's kind of like cancer. Your body is really smart and it figures out workarounds, unfortunately.
Wait, one last question.
Okay.
In line with the theme, autoimmunity in 2025, what's your big pick for 2025? What will be the biggest new development between now and 2025?
Well, one area that I see as being really valuable is really, using the tools that we have now, the ability to look at individual cells, the omics data, and starting to understand the heterogeneity of the disease and our ability to stratify patients. And also, by understanding the heterogeneity of the disease, it may also point us to new targets. I mean, this has been huge in cancer in the last 3 years, is, sequencing different kinds of tumors and realizing, oh, there's this other kind of, brain tumor, and we can use this drug. It's already FDA approved. And, I think repurposing drugs that are being used for other immune disorders, so you don't have to go through 10 years of FDA approval and, preclinical testing. I mean, I think that's another big area is repurposing drugs that are used for other diseases. So I really see us, kind of moving the therapeutic pipeline and honing it more specifically to the patients who are most likely to benefit is really where we're moving forward.